This is the lecture for Chapter 19, Disorders of the Immune System, Part 3. Again, with the disorders of the immune system, there are two general types, an overreaction and an underreaction. So far in these lectures, I've gone over overreactions or hypersensitivities. I've talked about allergy, which is a response to a foreign non-pathogenic antigen called an allergen, and in autoimmunity, where the immune system is responding to a self-antigen. There is one last type of overreaction that I'm going to go over, and that is transplant rejection. Transplant rejection is where your immune system rejects or fights against transplanted tissue. So tissue transplanted from another individual. And the main mediator of transplant rejection are the CTLs. Because remember, CTLs, their main purpose is to kill eukaryotic cells that should not be in your body. When CTLs are patrolling your body looking for eukaryotic cells that are foreign to your body, they are mainly looking at certain molecules called MHC. MHC stands for Major Histocompatibility Complex. So that's the name for them in mice. In humans, the same molecule is called human leukocyte antigen. Because remember, the CTLs are patrolling your body and they are actually looking for antigens that would activate them. So these are the human leukocyte white blood cell antigens. And these are molecules that you find on the surface of the cells. They are glycoproteins. They are part of the human glycocalyx. Human leukocyte antigens are found on the surface of all your cells. There are three major antigens, HLA-A, HLA-B, and HLA-C, and three minor antigens, HLA-E, F, and G. For HLA-A, there are 1,884 different options. For HLA-B, there are over 2,000 options. For HLA-C, there's over 1,000 options. These HLA molecules are encoded by genes, and so you got one gene for HLA from your mother and one from your father. So possibly for HLA-A, from your mother, you could have received the gene for HLA-A number 500. And for your father, you could have received the gene for HLA-A for number 1387. So you receive your HLA genes from your parents, and then that combination is a unique combination to you. When receiving a tissue transplant, in order to prevent rejection, you want the donor tissue to match your HLA as closely as possible. The closer the match, the less likely the tissue will be rejected. An autograft is one type of source for tissue that can be transplanted, and the auto tells you where it comes from. An autograft comes from yourself. So tissue that is taken from your body and put into another part of your body would not be rejected because it has exactly the same HLA. The only problem with an autograft is that the only transplant that can be autografted, or the tissue that can be autografted, is skin. So if you have a burn on one part of your body, you can remove skin from another part and cover the damaged area. And that skin should not be rejected because it has exactly the same HLA, so your white blood cells will not recognize it as foreign. So autografts work well for skin transplants, but for any other type of tissue, like a kidney, you can't use an autograft for that. So instead of an autograft, you would have to look for a source elsewhere. An isograft would be your next best option. And an isograft is when tissue comes from your identical twin. And identical twins come about because one fertilized egg at some point in development divided and produced two individual people. And because those two people came from the same fertilized egg, that means they have exactly the same genes, which means that they are clones, genetically identical individuals. So because they are genetically identical, they have exactly the same HLA molecules, so that tissue would not be rejected. If you don't have an identical twin, then the next option is to look for an allograft, and an allograft comes from another person. And when an individual needs a tissue transplant, like a kidney transplant, the first place they look at are that person's siblings, because you have the highest probability of sharing HLA with your siblings. Within a family, an individual child can only share a maximum of 50% of the genetics with either parent. And that is, of course, because your father donated 50% of your DNA to you and your mother donated 50%. So in this case, if this is you and those are your HLA types, half of your HLA came from your father and half of them came from your mother, which is true for all of your siblings. So depending on how the genes arrange themselves in your siblings, you may match 100% with a sibling, or you may match 0% with another sibling. So that's why when you need a tissue transplant, they go to the siblings first, in case they can find an individual who matches 100% with you, and that would reduce the chance of tissue rejection. If you don't have a match within your family, then the next source of an allograft that would be looked at would be the larger population. And this is where you go and look at potential donors, and if there's somebody willing to donate a kidney to you, they are tissue typed to see how well their HLA matches with yours. And again, the closer the match, the less likely there will be rejection. The last type of graft is a xenograft, and a xenograft comes from a different species. And the one species that is the most commonly used for human transplants are pigs. The internal organs of pigs are arranged in a very similar way to our internal organs, and they also have about the same size. So there are actually xenografts that use tissue from pigs and they're transplanted into people. Normally, this would cause a huge amount of rejection because, of course, the HLA would be very, very different on pigs than humans. But there are special sites in the human body called privileged sites. And privileged sites are areas of the body that are not under the immune system. They're not regulated by the immune system. Antibodies don't go there. The CTLs don't go there. So they have no immune components monitoring those areas. And some privileged sites are the cornea. If you get a corneal transplant, they don't have to do a tissue type. You can get a corneal transplant from anyone. There are also the heart valves, and heart valves often can come from pigs because, again, the antibodies and the CTLs don't go there, so there's no tissue rejection. And the last privileged site is the brain. So the brain, you've heard of the brain-blood barrier, where drugs and certain things don't cross from the blood into the brain. Well, that is true for the immune components. Antibodies, complement, CTLs do not cross from the blood into the brain. So the cornea, the heart valves, and the brain are privileged sites. A bone marrow transplant is a type of transplant that is very different from a solid tissue transplant. And a bone marrow transplant is used to treat conditions of the formed elements of the blood. So the formed elements of the blood, remember, are red blood cells, platelets, and the white blood cells. So when an individual has a problem with any of those formed elements of the blood, red blood cells, platelets, white blood cells, so it would be like leukemias, lymphomas, that means there's a problem with the stem cells in the bone marrow. So a bone marrow transplant is where you are replacing those stem cells with stem cells from another individual. So when a person gets a bone marrow transplant, this is the recipient getting a bone marrow transplant. So this is a person who has a problem with lymphoma or leukemia. First thing they do to the recipient is they have to get rid of those defective stem cells. So they give the person anti-cancer drugs, chemotherapy to kill the stem cells, and also radiation to kill the stem cells. So they need to eliminate that person's defective stem cells. Then they find a healthy donor that hopefully matches HLA. They remove some of the bone marrow from the pelvis. And then they can use FACS analysis to separate out the stem cells, if you remember how to do FACS analysis. Then they take those stem cells and introduce them intravenously into the recipient. And then those stem cells actually will home to the bone marrow, where they will start dividing and producing the red blood cells, the platelets, and the white blood cells. So in a bone marrow transplant, what you are transplanting are stem cells. And those stem cells are going to give rise to all the formed elements of the blood, all the red blood cells, the platelets, and the white blood cells. A major complication of a bone marrow transplant is graft versus host disease. So in a bone marrow transplant, you are transplanting stem cells that are producing all the formed elements of the blood. And graft versus host disease occurs when the white blood cells from the donor recognize the recipient's body as foreign, and they start attacking all the tissues. And that is graft. The grafted white blood cells are attacking the host body. And again, that happens when the HLA is not matched well enough, so that the white blood cells are recognizing all the tissues of the recipient's body as foreign. Graft versus host disease can have different levels of severity. Here there are two levels. Early acute GVH can cause a rash on the skin, and more advanced you can have a lot of damage being done to the skin. Again, this would involve the CTLs that are just destroying the epithelial tissue. This could also involve CTLs attacking the intestines. So early you have a little bit of damage, and then later with advanced you can have more necrosis. Other parts of the body that can be targeted for GVH, graft versus host disease, you can have the skin that is attacked, also the liver. So the liver can be damaged, and you can have lymphocytes, white blood cells going into the liver, and also the colon can be damaged. The mucosal surface of the colon can be damaged, and those cells can be induced into apoptosis. And that would be damaged mainly by CTLs, but also antibodies could be causing this damage. And for graft versus host disease, the only treatment is immunosuppressive drugs to reduce the action of the immune system. So far, I have gone through overreactions and hypersensitivities in terms of allergy, autoimmunity, and transplant rejection, where the immune system is responding when it shouldn't respond. The last thing I want to talk about is underreaction. Underreaction when your immune system is not responding when it should respond. And remember, your immune system is mainly responsible for protecting you from cancer and from infection. Immunodeficiency. Immunodeficiency means the immune system is deficient, not functioning the way it should. And one of the major consequences of being immunodeficient is the development of cancer cells in the person's body. CTLs, remember, are responsible for eliminating any eukaryotic cells that should not be in your body. And one example of that is the cancer cells. When they identify a cancer cell, they will interact with the cancer cell and induce apoptosis so that the cancer cell lyses and dies. And that's normally what CTLs do. If your CTLs are not functional, then there's no way for your body to eliminate the cancer cell, and the cancer cell will continue to grow. It is assumed that each human is composed of about 1 trillion cells, and it is also assumed that every day you generate 10 potentially cancerous cells. And when cancer develops, it starts here with normal cells. You have normal cells, and maybe one of the cells makes a genetic mistake, and it evolves into a cancerous cell. And so that cancerous cell will start replicating, so that one cell keeps replicating. And these are pre-cancerous cells, and that can progress as the cell continues to replicate. And then that further progresses into cells that now are not functioning normally, a lot of abnormal growth, and that would be a tumor. And then an invasive cancer would start spreading into other tissues. And then when it metastasizes, that means it gets into the circulatory system, into the lymphatic vessels, and spreads to another part of the body. So normally, in a person with a normally functioning immune system, the CTLs would eliminate that cell before it progressed into cancer. But if you are immunocompromised because you're very old, or you are a smoker, or you are immunodeficient because you acquired a disease, then the CTLs will not eliminate that cell, and this process will progress. An acquired immunodeficiency is a condition where a person had a normally functioning immune system, and then something happened, and they became immunodeficient. And the most famous example of this is the virus HIV, human immunodeficiency virus, which can lead to the disease AIDS, acquired immunodeficiency syndrome. And this was part of how they identified AIDS and the epidemic of AIDS in the 70s. They noticed that young, seemingly healthy young men were developing a cancer called Kaposi's sarcoma. So it's a very unusual cancer that would only develop in immunocompromised people, and they noticed basically an epidemic of this, so they started looking for a reason that those individuals had acquired an immunodeficiency, and they found the HIV virus. Immunodeficiencies can also be inherited, so those are congenital immunodeficiencies where an individual inherits a defective gene that has made their immune system compromised. The most famous example of this is SCID, severe combined immunodeficiency, and there was a famous boy who had it, and his name was David. He was born in the 70s, and he's the boy in the bubble. So when he was born, they identified that he had the mutation that gave him no functional immune system, so they raised him in a sterile environment. And he was in that sterile environment until about the age of 14 when he had a bone marrow transplant from his sister. And it worked, but unfortunately he developed cancer and died at about the age of 16. Inherited immunodeficiencies are very rare, but there are several different types. So severe combined immunodeficiency, that means both the T cells and B cells are affected. So the kind of SCID that David had was X-linked, severe combined immunodeficiency, so a gene on the X chromosome. And what that prevented, it prevented his pre-T cells and pre-B cells from forming. So he was unable to form any T cells or B cells. Another type of immunodeficiency is DiGeorge syndrome. DiGeorge syndrome prevents the pre-T cells from migrating to the thymus, so they cannot undergo T cell education. They can't mature, so the individual ends up with no functional T cells. Another type is ADA, where the T cells mature, but they're unable to be activated. So they're unable to be activated, so you have no functional mature T cells. There are also disorders that can affect just the B cells. Congenital A-gamma globulinemia prevents the B cells from maturing. So without mature B cells, you cannot have them responding and producing antibodies. Also, there is another one, hypogammaglobulinemia, which prevents the mature B cells from being activated. And without activation, there will be no antibody production. And individuals who are unable to produce antibodies will be susceptible to recurrent bacterial and viral infections. So as a review of the immune system, it would be a good idea for you to think about different types of congenital immunodeficiencies and what effect that would have on the individuals in terms of fighting infection and potential disorders they might have. So remember a couple of the important disorders. SCID means there are no T cells or B cells produced. So you would have no cell-mediated immune response and no humoral immune response. DiGeorge syndrome would prevent cell-mediated immunity. And then congenital A-gamma-globulinemia or hypogammaglobulinemia would have no humoral immune response. So you should know how these different disorders would affect an individual's immune system and their response to different types of infections and also different immune disorders.